This conference will now be recorded.
Okay, and I'm going to go to the slideshow and have it be from the current slide. Very good. Okay, so folks, so welcome to the class this evening. Thank you for coming. We are going to be looking at the study of tissues, histology, and looking at some very interesting slides. And really, this will just add to some basic information that I shared with you all in the labs for lab number 1 for both groups A and B. Whoever has their phone, let's see, because I need to let me stop sharing for a moment here to see what's going on. All right. Okay, very good. Thank you for okay. Share back the screen. Excellent. Okay, very good. So we're looking at study of tissues here. We're looking at this histology. Histology, I have to tell you, each semester it can be very challenging, right? It's not the easiest thing to review and to study, and many of you are not really very familiar with microscopes, although some of you have some microscopy background as far as in high school or in other college classes and such. This is why I take the time to really set up the microscopes for you all. I put them in focus and I show you what I'm looking at, because to me, it's going to be a waste of time for you to have to really try and search for in those slides and find the correct examples of the tissue. Because know that the slides that you're looking at have examples of multiple tissues, not just one tissue in particular, even though it might say on there simple squamous epithelial tissue for the tissues on the slide itself, as far as a marker and identifying piece of little tape there. But know that really it has multiple tissues present. And so I want to get through and have you not wasting time and focus on the fact that, hey, here's what it's looking like. And you all are going online and doing your Google images and looking at examples of these tissues under 400 magnification, as well as I said to you that look at them also in 100 magnification, but primarily it's 400 that you'll be tested. There's one that I might test you on 100, and that might be if you're looking at spongy bone, because you're able to really see the trabeculae, the sponges, the rods of bone that make up that lattice work of bone in spongy bone. So we'll go from there. But that's primarily, it's really 400 magnification, 400x. Okay. So now, as we're looking at this here, you see these two terms that I'm sure you're familiar with. You're familiar with biopsy. You're familiar with autopsy. No, in the study of medical terminology and what we do is through the medical profession, we're using these terms and such, that many times terms will have multiple AKAs, also known as. So necropsy, autopsy, AKA, same thing, right? Also known as. And so we'll see here as far as tissue collection, whether it's tissue collection from living tissue or dead tissue, necrotic tissue. That's what we're looking at there as far as being able to see as far as in an autopsy. Hey, so what was exactly what the patient had died from or possibly contributed to their demise. And the biopsy, hey, we're just trying to find out whether we have a cancerous condition going on or whether the condition is not non-cancerous and we can safely treat it, or if it is cancerous, then how do we specifically treat that cancer? So epithelial tissue, right? So when you think, so you have to, in your mind, when you hear about the 4 groups of tissues, right? So we have epithelial tissue, we have what? We have then connective tissue, muscle tissue, and then nervous tissue. When you think about those 4 groups, the specific function or what goes or how you correlate and differentiate between them needs to come at you right away. So in epithelial tissue, I'm thinking that 1, it covers and lines structures within the human body, and 2, I think avascular, that there's no blood vessels present in epithelial tissue, so avascular. Know that cartilage, which is a connective tissue, is also avascular. Also, something that you can keep in mind would be that the cornea, so you have the white of your eye, right? The white of your eye is the sclera. The cornea is the clear portion of your eye, avascular. And also the lens present deeper within your eye helps to refract light in order onto the posterior aspect of the retina. That's also avascular. It's clear. Okay, so you need to know some structures that are avascular no blood vessels present. And you just see here as far as role and functions and such. And so really covers and lines, very important. Absorption of nutrients, right? Because if there's one layer, if there's a simple layer, it will be easy to have nutrients, to have gases pass through it, okay? As far as glands are concerned, also epithelial tissue, secretion of mucus, enzymes, sweat, right? Oil, hormones. Now know that we have endocrine glands that will secrete the hormones. Exocrine glands will secrete the mucus, the enzymes, the oil, the sebaceous glands, the sweat glands. Okay, so there's different types of glands also, and then excretion of waste. Now, I mentioned to you that in lab there, I showed you an example of a columnar cell, and I said at the top surface, that free surface, the top area of a cell. So when we're looking at the columnar cell, you'll see that at the bottom portion, right? I'm just using some simple terms first. The bottom portion of the cell is going to have the nucleus, right? That would be closer to the basement membrane, the basal layer. Whereas the apical surface, the very top, the free surface of that cell will have the exposure to the lumen. So when we think of a lumen, so I have my paper towels here, and you see the inner hole area, right? So say that this represented whether it was a digestive system structure or a blood vessel, right? The lumen is the inner portion of that structure, okay? And so epithelial tissue will cover the line. So cover the outside of the organ, cover the inside of the organ, okay? Avascular, we talked about. And really, what goes on with epithelial tissue is that it's able to replace damaged cells very quickly. So it's highly mitotic, okay? So mitosis, right? Mitosis is cellular division. So know that if we're thinking of, so let's step away for a moment and think regarding cancerous conditions, okay? And say we're receiving chemotherapy in order to handle a cancerous lesion within the body, right? What kind of symptoms might it cause for you all? So let's stop sharing for a moment here. Let's show everyone, okay? So what do you think as far as, so I have everybody's viewed right here, right now. So you tell me what you would think as far as from experiencing chemotherapy, what might be some of the symptoms that a patient might experience as a result of going through chemotherapy. So let's hear someone. So Norma, go for it. I can't hear you. That's okay.
Loss of weight.
Okay. All right.
Hair.
Yeah. So that's a good one because of the fact that really like the tumor or that cancerous lesion is actually removing a lot of nutrients that should be going to other parts of the body. Good. Okay. That's good.
Loss of hair, loss of weight,
okay.
Loss of hair.
Okay. Good. Loss of hair, loss of weight. What else?
Feeling frequent fatigue.
Fatigue, yeah.
Okay, so fatigue. All right, good. What else?
Bleeding.
Bleeding.
Loss of appetite.
Okay. Okay, issues with digestive issues.
Yeah. Also tissues, like it's harder to replace the cells.
Good. Good. And so we'll stop right there. That's very good. Let me just see who put something in the comments, but hard to replace tissues. So what I'm going to tell you, right? So let's just see who. The immune system compromised. Sure, yeah, because really we're really the chemotherapy is just destroying structures, right? So it's not only just destroying the cancerous lesion, cancerous tissue, the tumor, it's also destroying healthy tissue. And so here's what I want you to think. So if cancer cells are rapidly dividing cells, right? Because they're usually, but not all the time, but when we're thinking of rapidly dividing, we're growing bigger, the tumor gets bigger, right? So think about what area of the body do we have mitotic division, major mitotic division taking place on a very quick basis, replacement of cells. We think the epithelial tissues. So we think that your hair, how about your nails, right? your digestive tract, so nausea and such, right? These are all areas that epithelial tissue is present and can be damaged and destroyed. And so even when someone said about replacing tissues, absolutely. So you're affecting these tissues in such a way so this contributes to the symptoms. And we do have medications now that are much better than they were years ago that can help to manage these symptoms, these side effects as a result, right? So I want you to understand that so you can see that, hey, if it's the chemotherapy, and they're trying to develop chemotherapy that's more specific for the specific cells and such, but it's kind of tough. So there still can be, like, you know, other areas of the body that can be affected. And again, if it's attacking rapidly dividing cells, epithelial tissue is a target. Okay. Any questions or comments before I go back to the PowerPoint? We good? Uros, you good?
Yes, sir.
Okay, so please mute so make sure you mute, good. All right, I'm gonna hide everyone and go back to the PowerPoint. All right, so let's move on here. So also name according to shape, arrangement, function, right? So that's very important. And so we're gonna look at that. We've already talked about this, but just as a review. So as far as shape is concerned, so we have those squamous cells, we have the cuboidal cells, and we have the columnar cells, and we're very familiar with what they look like. And so you should know, right, not only just the shape, but what's the action as far as what is the function of these specific shapes for the epithelial tissue, okay? Because if you know this, then you can have an idea where they might be located, these specific types of shapes and of the epithelial tissue. So here we have squamous epithelial cells, okay? And so know that, so we have here, right here, right? So you're looking at simple squamous, okay? So you're looking at multiple layers, but see, as we get to the apical surface, right? You're seeing here, right? So over here, this would be basement layer, right? So here we would have more of the apical surface right here. Okay? And they're flatter cells. Those squamous cells are flat cells. Okay? When we look at, like say the esophagus, we would look at those apical surface, those outer, the outer cell for multiple layers. So this is just, so this would be stratified squamous, okay? But simple squamous, we would just looking at one layer, multiple layers stratified, okay? And know that when we're looking at those, the apical surface, the very top layer of the stratified layer, we're looking at the flat cells, okay? And they're going to be, if it's the in the esophagus, they'd be alive. They would have a nucleus present. In the case of skin, in the epidermis, that outer layer, that apical surface layer, would actually be filled with keratin, and so we would say that that's keratinized, and it's no, those cells in the outer surface are dead cells. Cuboidal epithelial cells, right? So here you're seeing these cube-shaped cells, right? Here's the lumen, okay? And so simple enough. You know, just looking at the cuboidal, that's easy to spot. Also here, look at this as far as, now, can it look like there might be multiple nuclei? It's because, understand, when we're taking thin slices of the tissue, you can, it's more, it's not just extreme, it's not just flat. There's some depth to it, and that's why you're seeing where it looks like there might be multiple layers of nuclei, because you're just seeing the depth to the tissue, and that there are other simple cuboidal cells right behind the outer layer that you're viewing, okay? And you're seeing here also, this right here, we would call cilia, right? So cilia allows for movement of material along the apical surface. So here's the white is the lumen. So the apical surface is right here, the top layer of the cell. Here's the bottom layer of the cell with the nuclei, and we'll have the basement membrane, which will be anchoring it to the deeper tissues, okay? The connective tissue that's at that basal surface, that bottom layer. So simple, one layer, stratified, more than one layer, pseudo-stratified, appears to have more than one layer, but it does not. And then transitional does have multiple layers, so it's stratified, but also know this, that at the apical surface, at the very top layer, those cells are more oval in shape, okay? And they allow for stretching. So think the urinary system, and think in particular urinary bladder. This would allow for the bladder to expand and then reduce back in size once the patient has voided. Now here we have pseudostratified ciliated. So pseudostratified appears to be multiple layers, but it's not. And you're going to see cilia present at the apical surface at the top layer. Okay, here's the basal surface. Okay. And it does look similar to, but it does look different than the columnar. So let's look at columnar again. See the columnar? There's definitely more. It's similar, but it's different. And again, too, reviewing multiple images will help as far as reviewing it over and over. Histology World is a very good website. There are all kinds of websites you can look at, but Histology World is a good one as far as some quizzing and testing that you could do also to help you learn the material. Transitional. So look at this apical surface here. Do you see at the apical surface, right? It's multiple layers, is it not? And you'll see that we'll have oval shaped type cells at that apical surface over here. So I try to give you the best possible example I can when it comes to testing time if I'm choosing a transitional, that you see that at the apical surface. So what's the white? The white is the lumen. So at that apical surface, we have the oval shaped cells, multiple layers, transitional epithelial tissue. Like I said before, the non-keratinized and keratinized. Non-keratinized, we have live cells at the apical surface of stratified epithelial tissue. Keratinized, we're seeing as far as the skin, the epidermis, will have these cells that are dead and they're filled with keratin, a protein, okay? And so they allow for this durable, dry, moisture-resistant appearance and function, okay? The oral cavity, the esophagus, live cells, they're moist, right? Really, these are dry as far as the keratinized, the dead cells, okay? Live cells need moisture. If they don't have moisture, then they're going to die. And so hence, the epidermis is full of these dead cells, keratinized epithelium. I talked to you about this earlier there, and I mentioned about exocrine and endocrine glands. So exocrine glands, we think, you see there, sweat glands, we think mucus-producing glands, salivary glands, mammary glands. These are all producing substances that are going to be released out of the cell, okay, and going out of the body and such. Oil-producing glands, sebaceous glands, exocrine glands. The endocrine glands are producing hormones. These are chemical messengers that are sent into the blood, into the cardiovascular system. They're produced in one area and they go to another. Next semester, we'll be looking at that in the beginning of the semester. As a matter of fact, your AMP2 colleagues are looking at endocrine slides, endocrine histology. So know that you don't really get rid of histology, okay? So you'll still be looking at slides and samples of slides and such, okay, in AMP2, but it's not as intense as AMP1 is, right? So these are also a method of secretion that we can label the glands, and you'll see here as far as merocrine, apocrine, holocrine. So when we think the merocrine, we think that only the substance is being released by this gland. Apocrine, think in terms of there's a piece of the gland that's actually going to be released into the secretion, okay? So hence the AP there, see the AP, apocrine, a piece of the gland will be released into the secretion. And then holocrine, whole cells can be a part of the secretion. Whole cells are part of holocrine secretions, okay? And you'll have to know the examples of. So there are also, notice that there are also apocrine sweat glands, and we would say that they would be present in the axilla, your armpit, and also the pubic region. So you see how the AP also comes into play for apocrine. So primarily the sweat glands on your body release a very watery type sweat that would be all over your body, except for in the case of and after puberty, really your axillary region, your armpits, and also your pubic region and such. These would have more of a oily type of sweat, and really bacteria like that, and so that can produce a little bit of a smell also. So just is what it is. Okay, connective tissue. So know that connective tissue, we're not going, oh, by the way, so let me just, I want to review something else for you here. So look at these tissues. Are these tissues tightly packed? They are, right? So epithelial tissue has tightly packed cells. Now, when we get to connective tissue, know that we're going to have a matrix in which cells are present, but the cells are going to be interspersed and kind of spread out a bit. They're not as close together, per se, in connective tissue as they are in epithelial tissue, okay? So the cells are located further apart from one another. They're present in this extracellular material, this matrix, and many different functions, and we're going to look at them. So know that you have to understand a little bit as far as the structures that are making up this matrix. And so we have protein fibers. Collagen fibers are thick, strong fibers. Elastic fibers have this ability to stretch, stretching and or compression, and they can return back to their shape, and they're thinner than the collagen fibers. And then reticular fibers are even thinner than the elastic fibers. When you saw the example of the areolar connective tissue in the lab, you saw tissue that contained thicker collagen fibers that were kind of like pink purplish. They were thick. And then you also saw elastic fibers, which looked like I took a pen and I just drew some lines on the cell there on the slide. That would be the thin elastic fibers. Okay, all of them protein fibers, very important in the matrix. Also present within this matrix, right, we have the substance called ground substance, and this would contain substances that allow for the liquid portion of the matrix, right, to contain structures that will allow for and act as like a sponge, okay, also make the portion of the extracellular matrix very slippery. So if you've ever heard of hyaluronic acid being offered as far as a supplement, right, for folks that are having issues with their joints as they get older and such, very super slippery and very important to be a part of the substances present within structures where their joints are present. Proteoglycans act as a sponge, so they entrap large amounts of water, which is very important, and adhesive molecules present in order to keep structures together and keep the tissues together. So we have different types of cells, right? So fibroblast is a type of cell that would produce fibers, right? So we've talked about collagen fibers, reticular fibers, and elastic fibers. We have these different stem cells that will actually be able to differentiate and produce other types of cells. We have macrophages, which are present in your immune function and present within tissues that have an immune and lymphatic function that really work to patrol the body, looking for cells that are non-self, as well as cells that are dying. So macrophages are, we would say, are like the cleanup crew. They are involved in phagocytosis, hence the term macrophage. They're a big phagocytic cell that they're involved in cell eating. I think of it like a Pac-Man, if you ever recall that old video game or have seen that at all, but that's what I think of a macrophage. Kind of clicks for me. A leukocyte also is a white blood cell, okay? And these white blood cells also have phagocytic abilities and functions, and so they are also very important. Red blood cells, erythrocytes, right? We're not seeing it present there. Mast cells involved in immune function, and these fat cells, adipocytes. Just examples of some cells present within connective tissue. And remember, so we're saying, oh, well, leukocytes, and know that a macrophage began its life as a leukocyte, as a white blood cell, a monocyte in particular that differentiated to become macrophage. And also know that, recall that blood is considered a fluid connective tissue. So we talked about areolar connective tissue. I showed you that in the lab. It's collagenous fibers, loosely arranged, right? So you saw thick fibers, you saw thin elastic fibers present, and you saw cells interspersed in between. Very easy tissue to pick out and to identify as that, hey, I know that one, it's areolar connective tissue, because it's very unique in its appearance. We talked about dense regular and dense irregular tissue when we were in the lab, and we looked at dense regular as far as ligaments and tendons. Go back. As far as ligaments and tendons are concerned, recall that when we saw dense regular, it had that wavy appearance, right? Dense irregular, I talked to you about and showed you that we see that present in the dermis region. So we have the epidermis, the outer layer of skin, then which can be 4 or 5 layers itself. Then we have the dermal layer, which is the really connective tissue layer that anchors the epidermis, okay? And this dense irregular tissue also contains blood vessels. It's vascular. So this is important in order to allow for nutrition to reach the cells. Now, we'll talk more about this next chapter, okay? So just let you know that. And you're just seeing here other types of tissues and such we'll be looking at. So loose connective tissue, so we're thinking the areolar connective tissue, fills spaces in between structures and penetrates organs. This is important. It's that packing tissue, that areolar connective tissue. And it can contain all these different types of cells, as well as collagen fibers and elastic fibers. So here's that areolar connective tissue. Very, very easy to spot and to see and say, hey, that's areolar connective tissue. You'll see above, it's showing you here that we have these different types of immune cells, plasma cells. Are these plasma cells, interesting, they will differentiate and become, and well, actually, they started out as a type of B cell. They're B and T lymphocytes. These are specific types of white blood cells. They'll differentiate and become a plasma cell, which will produce antibodies, which are involved in the immune function. So you'll learn about that more next semester. So you're just seeing here all that's involved in the structure of areolar connective tissue, also known as loose, because doesn't it's very loose. There's a lot of these fibers and the cells are interspersed between. The dense regular and irregular. I recall I mentioned it, let's see, on Monday in lab, and I believe I did the previous Monday, but if I didn't, I'm going to say it here in class here, that the dense irregular, interesting how those protein fibers are in many different directions. So when we think of your skin, you could move your skin in all different directions, right? And it's still going to stay attached to that dermal layer, that deep layer of the skin. And that's important, so because we have these protein fibers holding the skin in all these different directions in order to keep a nice tight hold on the epidermis. And so here you're seeing as far as the dense connective tissue, right? And I give you a better example when you're in the lab, but wavy type tissue. Adipose tissue, we're thinking fat, right? So in the lab, I showed you a model of the skin, and I showed you at the hypodermis, the subcutaneous layer, aka the subcutaneous layer, the very deep structure, deep layer that is not when we talk about the skin, we're talking about the epidermis and the dermis. When we talk about the subcutaneous layer, aka the hypodermis, it's a layer that is supportive of the skin, but it's not considered the skin. But it does contain a high amount of adipose tissue. Know that adipose tissue contains these adipocytes, right? When I show you adipose tissue in the lab and to identify it and such, I will actually have the pointer pointing at the inner portion of the cell, so that this way you don't get it confused with a simple squamous epithelial tissue. Okay, I think of like soap bubbles when I think of adipose tissue. Soap bubbles, very easy to identify and think about it that way. Reticular tissue, think about the lymphatic system. The lymphatic system is involved in the immune function of the body, okay, of the body there. And so reticular tissue, these very fine collagenous fibers, I don't show you that in the lab, okay, but you have to know that what it is as far as for lecture exam, for test number 2. So all of this chapter is test number 2. So test number 1, again, remember, it's the introductory chapter. There's a lot of information there, a lot of terms. Then we have the chemistry, that handout that I gave you, and then chapters 3 and 4, which is the one PowerPoint combined that has the cells and also mitosis and such, cellular division. Okay? Cartilage. Recall that I talked to you about cartilage, and cartilage contains chondrocytes, right? Chondrocytes are cartilage cells. Chondrocyte, cartilage cells, okay? They're found in the lacunae, and you'll see in a moment here. Let's see. Okay. I don't have it posted here as far as in the PowerPoint, but I've showed you in the lab, and you can look at it, and I can, if you have any questions, I can find some images to post in here also regarding examples of hyaline, fibroelastic. But know that, again, the external ear, elastic cartilage. Recall the epiglottis, that's the, what, kind of a weird thing, okay? So don't judge me for saying this, but with the epiglottis, I think of like a toilet bowl lid, right? Because it just kind of reminds me of that, but it really does. It creates a seal, and so that seal that it creates really prevents any liquid or solid material from entering into the respiratory system, right? Into your lungs, and that's very important. So that's also elastic cartilage, as well as I said to you that the eustachian tube, the auditory tube. When you pop your ears, that's what you're using as far as your eustachian tube in order to equalize the pressure present within the middle ear. Fibrocartilage, toughest cartilage. I said to you that fibrocartilage, we're looking at the meniscus of the knee joint. We're looking at the intervertebral discs, your discs, fibrocartilage. When we also look at the TMJ, fibrocartilage, okay? The pubic symphysis region, where the pubic bones come together, articulate, fibrocartilage. Everywhere else, hyaline cartilage, okay? And so hyaline cartilage is really the structure that will become bone from the fetus, right? So the fetal skeleton starts out as hyaline cartilage and then ossifies, becomes bone, except for the ends of bone, which will continue to have that cartilage present, so that bone's not just rubbing on bone, but we have cartilage present in order to cushion the bone and protect the ends of the bone, as well as your growth plate it's called the epiphyseal plate or the growth plate, AKA. This is also hyaline cartilage. And we said avascular, right? Heals slowly. So when it does tear, meniscus tears are common. When it tears, slow healing. Bone, okay? So osseous tissue, okay? We looked at in the lab compact and spongy bone. Now you need to know that compact bone is also known as cancellous bone, okay? There's also another term here, and let me put this in you should write this down, please. Trabecular bone, okay? Trabecular bone has those trabeculae, right? Those trabeculae, those rods of bone, okay? And allowing for, so it's not just all solid with the compact bone, right? It's these trabecular, these spongy, it has holes present because of the rods of bone intertwining, but having spaces between, okay? And we're gonna be talking about the next chapter will be the integument. The following chapter we'll be talking in more detail regarding bone. But again, know that bone has a rich blood supply. So when bone fractures, you bleed, right? And so know that bone is highly vascular, and bone will produce, it's hemopoietic, it will produce blood cells, okay? Very important. And here we're looking at, what is this an example of, right? It looks like a tree trunk that's been cut. This is it's called an osteon. It's a functional unit of the bone. We'll talk about that in skeletal system chapter. This would be compact bone. The blood, we talk about blood, that's a liquid, fluid connective tissue, right? Very unique as far as the connective tissues are concerned. The liquid portion, we would call that the plasma, and the formed elements are the cells. You need to know these terms. The plasma is the liquid portion, the formed elements are the cells present. And so I didn't mention to you this term, platelets. We're also, go back, and I'm going to also, platelets are also known as, so I've talked about red blood cells are erythrocytes, white blood cells are leukocytes, platelets are thrombocytes. You need to know that term. And we'll be looking more at the cardiovascular system in A&P2, okay? Let's see here. And so really the role, the function of the blood, transporting nutrients, gases, hormones, waste, removal of waste, and really involved in the immune function of the body. So this hemopoietic tissue is bone. It's able to produce the bone cells, the blood cells. And it's found in what in particular red marrow. Okay, so red marrow produces these blood cells. Right, yellow marrow, a storage area for adipose tissue. But know that with age, yellow marrow replaces much of the red marrow. Okay, so that we have, so that there's, you know, any type of function that takes place as we get older slows down and doesn't function as well. That's just the degradation. It degrades over time, right? But know that if there's a need in the body, the yellow marrow can become red marrow if there's a need for increase in blood cell production, say as a result of an injury, accident or injury. And also I mentioned to you all that the marrow is present in the ends of bone, in the epiphyses, as well as the cavity there, medullary cavity, okay? Connective tissue functions, simple
Sorry,
enough,
sir,
folks.
sorry, sir, can you repeat that again, please? What did you say about medulla?
So the medullary cavity, let me go back to that slide and I'll adjust it here. I'll give you the bone marrow and let's see, back up. The medullary cavity is the area that's the hollowed out portion. So when you think of, so look at my paper towel again, right? So we have here, so imagine that the paper towel itself, that's all compact bone. What lines the medullary cavity, the inner portion of the bone, would be spongy bone, okay? Spongy bone would be in the inner portion of that medullary cavity, and then marrow would be present there, as well as at the ends of the long bone. And it's red bone marrow that produces, it's hemopoietic, produces these blood cells. But again, that medullary cavity is the inner cavity containing the bone marrow, which will primarily be yellow marrow. Good? Okay. Very good. Also some more regarding storage and attachment, separation, right? Important. Muscle. So muscle, we know that there are three different types. There's smooth muscle, skeletal muscle, and cardiac muscle, right? So the smooth muscle, I mentioned you, it's these spindle-shaped cells. And remember that I said also to you that it really looks like the nucleus of a smooth muscle cell was stretched. And that's really the best way to identify that type of tissue, okay? Non-striated, and it's involuntary, we get that. And again, we're looking at digestive tract, we're looking at tubes, we're looking at blood vessels, ureters that contain the urine. This is what we're looking at as far as smooth muscle is concerned, involuntary. And you'll see to the right there, my viewing right, there we're looking at these nuclei that have been stretched. We're looking at these spindle-shaped cells. Skeletal muscle, we're looking at long muscle fibers, right? Long muscle fibers, multiple nuclei. There's striations present, right? There's that. You know, do you ever recall like a fabric that pants would be made out of called a corduroy? I don't know. That just comes to my mind as corduroy when I think of striations present within. I don't know. It just clicks for me. So, but it's these striations present, these lines present in the skeletal muscle that you're seeing as a result of really the very structural components of the protein fibers present within the muscle tissue. And we'll look at that more in a couple of chapters, okay? And you're seeing here, see the striations present, right? See those striations? You can see them present here. Multiple nuclei, multiple nucleate, right? And cardiac cells. Cardiac cells think that they are what? 1, involuntary, 2, striated, 3, there's branching, and really 4, you see these intercalated discs, okay? So let's take a look at that and just, it'll be very helpful to see. Yeah, you see those dark lines here? So you see the striations, okay? We see that there's branching, cardiac muscle. We also see this like a dark line here and there. And those are those intercalated discs present and allowing for the communication between the cells within the atria and the ventricles in order to allow them to contract as a unit in order to maintain the dual pumps, right? The pumps for the pulmonary system going to the lungs, as well as pump for the systemic, pumping blood to the rest of the body. The nervous tissue, this is our last tissue, and so it's comprised of nerve cells, okay? So a neuron is a nerve cell. Primarily what we're going to be looking at would be, here we go, and I'll come back in a moment, but you see this example right here of a multipolar neuron. In other words, we have multiple processes, we have multiple dendrites, one axon, multiple processes, so it's a multipolar neuron, okay? And that's what we were seeing in the lab. So that image right there is really what we're seeing in the lab. That's what we're looking at, this nerve cell with all these support cells surrounding it, okay, in the nervous tissue. So there's a cell body. Also, you should know, we call this the soma. Soma is a term that you need to know. That's the cell body, okay? Contains the nucleus, contains the organelles, contains the liquid portion, the cytoplasm, the cytosol, which is, the cytoplasm is all the guts present within that cell body. The dendrites are actually receiving stimulus. I'm going to give you another term that I want you to know also regarding this. So they receive stimuli, so they receive, we would say, afferent information. You need to know this term, afferent. Put a little deal here. Afferent. And that know that the axons are EFFERENT. And that's a term that you need to know for this semester as well as next semester. So you just need to realize that afferent information is coming to the cell or it's coming to the nervous system. Efferent information is going away from the cell body, the soma. Or in particular, we could also use it as far as nerves are concerned. Efferent information is going away from, it's exiting the nervous system and going to some other part of the nervous system. Afferent, efferent, very important terms for you to know and to remember. So the support cells will be going into more detail at the latter part of the semester in our neurology, and we'll see that neuroglia, these are nerve cells, these are support nerve cells. So they're not nerve cells, they're supportive cells of the nervous system, of the neurons, okay? And there are different types. We have different types present within the central nervous system and the peripheral nervous system, and we'll discuss them in more detail. But know that they are supportive, like this says, they're supportive of these neurons. These neurons require a lot of protection as well as nutrients, as well as oxygenation and such. So very important to control, and also protection as far as allowing for a barrier between the body and the rest of the nervous system in order to protect it from pathology, from disease and such. But still, we can have issues within the nervous system, but when they bypasses, in particular, there's a blood-brain barrier. We'll talk more about that later on. And also talking about controlling emotion and reasoning, storing memory, perceiving. So we actually receive information. So we have receptors that will receive information and send it to the central nervous system via the spinal cord and then eventually to the brain. And then we'll have neurons that are actually sending out information away, that are sending efferent information away. Once the brain processes, the nervous system processes what's the input, then we'll have output, okay? And again, there you go as far as that multipolar neuron. There are other types of neurons. We'll talk about that more when we get into the beginning of neurology. So you see here that, folks, we have covered quite a bit in this chapter regarding histology and regarding study of tissues. Know that, you know, we're taking this really, the chapters now up until right now, this has all been foundational information. Now we're going to we're going to build our structure of anatomy and physiology upon the foundation of these multiple chapters here, so that when we come to next chapter, integumentary system, the skin, we're going to be getting more into detail regarding things and structures and such, and if the different body systems. Okay, let's stop our